I cannot shout so much. Just got back the voice and don't want to lose it again. um, is this for later? Right. Okay. We have only a few more subtopics left to discuss about. 9.8. Okay. All the subtopics learned from lessons last week. Do you revise the lesson last week? Or did you not? Fancy the quiz questions? Very easy. I can only be like a crazy lady, talk non-stop to make you do the revision. Learn, revise within 24 hours. There is no other way. Repetition is the key. There is no other way. When you learn in lecture, you listen. That is one sense that you are using. Within 24 hours, you read again with the notes that you have received. You use your eyes purely, not really auditory. That's another sense. So two senses come together, that will solidify long-term memory. That's how you learn. To have a committed and solid memory, understanding and everything, invoke all senses when you are getting your lessons. Okay? Right. So last week we learned about, in section 9.4, we learned about the organization of the photosynthetic apparatus. So this is where you understand that within the chloroplast itself, there are compartments, there are structures, okay? The important structures are what? What do you find in the chloroplast? Thylakoid, thylakoid, stroma. So thylakoids are the pancakes. These pancakes actually, in itself, you can further divide it into two. What is it? Grana lamellae and stroma lamellae. Right? Okay. These are very important because photosynthesis got various reactions. And these reactions happen in specific locations within the chloroplast. It's not some random biochemical reaction whenever the reaction decides to happen. It doesn't happen that way. Okay? And within this structure, you can see that there are important proteins. Okay? Hence, you learn the concept of integral proteins. What are the four main integral proteins that you learn? PS1, PS2, ATP synthase, and cytochrome B6F. Okay, that's good. Right. Okay, and 9.5, you learn about the organization of light absorbing antenna system. Okay, now you have understood that on the surface of the thylakoid membrane, whether the membranes within the region of grana lamellae or stroma lamellae, you have these islands of pigments patched all over the surface of the thylakoid membrane. And in each island, maybe 200 pigment molecules are present. However, there is only one reaction center. And this reaction center, which is a special chlorophyll A molecule, is surrounded by other pigments, and these pigments is the antenna complex. And bear in mind as well, these pigments are not standalone compound. They are always associated with proteins, meaning that some parts are in the hydrophilic region, a bit protruding out of the thylakoid membrane. Some of the structure is actually inside the membrane itself, embedded like an anchor, okay, in the hydrophobic region inside the membrane, right? Okay, so these collectively we known as LHC, light harvesting complex. Okay, no pigment stand alone. No, it cannot do that. Okay, because pigment on its own is actually pretty reactive, right? Okay, then what else did the children? I got my rota. I should be using this. Right. So in 9.6, okay, this is a bit a lengthy story. So you learn about the electron transport itself, meaning that you learn about the antenna system. This antenna system, once they have managed to capture the electron, get activated. By activated, I mean one electron has been ejected out of the chlorophyll and it has to go somewhere. If it do the photochemistry destiny, it will proceed to electron transport. The most talk about, the most, you can say, linear path of the electron transport, we call it as the Z scheme. Okay, because it goes up, gets activated, then go down the cascade of the electron transport, then it gets up again in the second photosystem, then it kind of go down again. What go up, what go down? The electrical potential of it. If you remember, there is a figure, it talks about the EM, mid potential uh, uh, electricity volt value of it. Right, okay. Then you also learned about the reaction center of PS2, which absorb optimally at P680. P stands for pigment, 680 stands for the wavelength, the amount of energy or specific quantum that can activate the ejection of electron out of the chlorophyll. For PS1, it absorbs maximally at 700 nanometer, right? And while this electron transport is going on in the Z scheme, other things are happening simultaneously as well. For example, one thing is the oxidation of water, and you need manganese ion for that. This oxidation of water, or the splitting of water molecule, is also called as photolysis. That's where you get your hydrogen ions and oxygen. Okay, two splitting of water, where you will get one molecule of oxygen. And 4 molecules of protons. When I say proton, I mean H plus. Remember, H can come in 3 forms. H plus, H minus, or H atom. Meaning that 1 proton, 1 electron. Right, okay. And then proton are transported into thylakoid lumen. Lumen is the inner side of the thylakoid, inside the pancake. When electron pass through the cytochrome B6F complex, look at the word complex here, meaning that these things, they are not a standalone structure. There are many subunits come together forming this electron carrier. Okay, remember again the concept of electron carrier and electron acceptor. Okay, most of the proteins that you find along the Z scheme, plastoquinone, plastocyanin, the cytochrome B6F complex, they are carrier because they actually literally move the electron to the next player in the Z scheme. But a compound like NADP, they don't really move things around. They just accept and they become something else, right? So that we call it electron acceptor, right? Right. So, and finally, NADP plus. What's the full name of this? How do you say this? The full name of this? N stands for what? Nicotinamide adenine dinucleotide phosphate. There's a plus there. So, yeah, you can see that. See nice. Yep. Meaning that it's lacking electron. It can receive electron. Okay. Nicotinamide adenine dinucleotide. If you might, sometimes if you um eat your food or eat something, get bored, you would read at the back of the label. For example, you are eating your gardenia bread or something. You can see in the list of nutritional profile, it says that nicotinic acid. That's the thing. This is actually a derivative of vitamin B3. Okay, it's the same vitamin that you get in your diet, right? The derivative of it. So that's why vitamin B3 is highly associated with energy production. This is all energy. Okay? You don't get enough vitamin B3, you are not going to have lots of energy metabolism going on. Right? Okay. And finally, in the PS1 reaction center, you will have these iron sulfur centers as the electrons carrier. Okay. What are these iron sulfur centers? There are three. What are they? There are three. What are they? Iron sulfur X. Iron sulfur what? After X. A. Iron sulfur B. Iron sulfur R. Where you found it? Cytochrome B6. The first route. There are two routes in the cytochrome B6F. The first route. Okay, right. And finally, you will find with the ferredoxin as the electron carrier. So in the ferredoxin, if it proceeds to have the electron get transferred to NADP plus, meaning you have completed the Z scheme. However, if the ferredoxin, the electron from ferredoxin do not go to NADP plus, but go back to cytochrome B6F, that's when you get cyclic photophosphorylation. Okay, so all this while, the pathway, the Z scheme, the other name for it is non-cyclic photophosphorylation. They are all synonyms. Okay, there are many synonyms for them. Non-cyclic, meaning that they are just only one way. Even though it's zigzag, it's still one way. However, ferredoxin, it can take another path. It can reverse back to cytochrome B6F. If it reverses back to cytochrome B6F, then the path is like in a loop. When it's in a loop, we call it cyclic photophosphorylation. Okay, I think the figure is helpful for this. It should be 950B, if I'm not mistaken. Let's see whether I remember that correctly or not. Because recently my brain got fried until 40 degrees. Some knowledge might have actually fallen down. Is it 915B? Figure. Is that right? Oh, aku tak gila lagi. You got to know. At least you know I'm not scamming. Ada kau tengok aku pegang aku. Kau patut ingat benda tu. Banyak tu pun tak boleh ingat. Okay, that's about it. Let's try to finish this off. 9.7, 0.8, 0.9. We're going to see how ATP is synthesized, how the photosynthetic machinery get repaired when it's too much happening to it, and the genome story inside the chloroplast itself. Okay. Oh, what's that? Oh yeah, photophosphorylation. You will see these terminologies from time to time. Okay, so make sure you have your little dictionary. I should make some kind of flashcard assignment or something to make sure that people actually not only memorize, understand all these terminologies. Okay, so proton transport and ATP synthesis in the chloroplast. So the formation of ATP, which is the energy currency, not only in plants but also in us, okay, is actually due to the concentration of hydrogen inside the thylakoid lumen trying to reach equilibrium with its opposite side. Okay, you have a membrane in the middle. One side has more chemical potential than the other. So when this happens, hydrogen will try to find a way to go to the other side to reach equilibrium. Why? Why it needs to reach equilibrium anyway? What's the fact? Why? What's something to do with hydrogen? Why is it? When a place is lost in hydrogen, what happens to it? It's too acidic. Okay, it's very acidic. So it's not only about the fact that the hydrogen is a lot there and it's too much to handle. It's like UPM, right? 25,000 students. It's not like some one night it tried to equilibrate. Some portion of student gets thrown into UKM. No, the students still stay in here, even though the portion of UKM, UPN student are not equal, right? So the reason is because of the acidity, and this will compromise the membrane integrity. Okay, will prevent the biochemical reactions that take place, and to some degree can denature proteins. Remember, there are so many proteins on the surface of the thylakoid, in the inner side of it. For example, the plastocyanin. Remember plastocyanin? It's on the inside of the thylakoid. Remember, if it's exposed to high acidity all the time, it's going to get denatured, right? Is that true? Acidity can denature protein. Give one example of protein denature. Give one phenomenon. You boil the egg. What happened to the egg? It's hardened. Why it hardened? The protein gets denatured. Okay? Denatured, as a result, the protein solidified. Okay? But that's heat. How we want to know how come high acidity can cause denature. Milk, milk, like goat milk or something. If you put some vinegar into it, you let it stir sometime, that milk can curdle up and you get solid as well. Okay, so kind of like that situation. That's why some people, they don't use the enzyme Rene to make cheese, but rather they use the vinegar to curdle up the milk, and then you got your cheese. Okay, no heat involved, no need. But sometimes they just put it on the warm stove, just to make sure they're not eating something not pasteurized. But actually, even if there is no heat involved, you still can get make curdle with the vinegar. Okay, right. So this actually want to talk about who found out that the um, chemiosmosis, that's the word, meaning that the achievement of chemical equilibrium between the two membranes, two sides of the membrane, is the one that actually drive photophosphorylation. Okay, in the case of plant, it is light, so you get ATP because of light. That's why the word is photophosphorylation. Phosphorylation means a phosphate group is added to something. In this case, phosphate is added to an existing ADP molecule in the stroma. In the stroma, you already have ADP, adenosine diphosphate. It's always there. So the addition of phosphate into ADP, why? Because ATP synthase has hate. Actually, you can understand this better if you read this web topic. It's in the folder actually. I have put in the folder. It should be web topic number. I forgot to put it on the slide. It should be web topic number 9 point something. Let's see. Keep forgetting the internet so. Is it live finding? Ya, yang harap lah kan internet kat sini. Oh yeah, this thing in here. So this is lumen, this is stroma, this is your membrane in the middle. Here you got your ATP synthase, one of the integral proteins. So ATP synthase got two parts to it. It is conventionally labeled as CF0 and CF1. CF1 here is the hydrophilic part of it, hydrophilic subunit, and then you have CF0, hydrophobic subunit of it. Okay, so you have your rotor head, you have the anchor body here, and then you have like a bridge to connect within these two structure. Right. What is CF? Stands for what? Actually, ATP synthase not only present in the chloroplast, it's also present in mitochondria. It is also present in vacuole, and sometimes it can also present in certain membrane. Okay, meaning that the present is kind of everywhere, anywhere throughout the living kingdom. Okay, so I think if you can see in mitochondria later, you only have F1 and F0. You don't have C. I think C, they use it to signify this is meant for chloroplast. Yeah, because if the ATP synthase is on the vacuole, they label it V1, V0. V to signify the whether it's a vacuole. Okay, right. So look at this thing. So hydrogen, lots of hydrogen, try to exit the membrane through the tunnel of the ATP synthase here. Right? How? This thing, the structure in here, this embedded protein here, so it can actually turn. Okay, this thing can actually turn. So every time it turns, three hydrogen can go with each round, can go up. then will eventually reach at the top, and at the top here you can see, so this is the aerial view of this rotor head. So this rotor head here, this is the aerial view of it. That's already an ADP. Okay, ADP here, and also the inorganic phosphate, the PI. So when this thing turn, it will jam the ADP PI incorporation with the phosphate. And the product is you get ATP, right? Again, in the beginning, there is only ADP and phosphate, loosely bound. But when the rotor head turn, ADP and P is jammed, is forced to fuse together. When they forced together, they will eventually form ATP. Why? Because the rotor head, it can turn. It can turn, it can squeeze things in. It's like you're wringing your wet laundry. Yeah. You get your ATP. This process of adding phosphate, inorganic phosphate to ADP, that is phosphorylation. Because this whole process, due to the movement of electron, because of light excitation early in the first reaction, in the PS2 reaction center, scientists add the word photo in the beginning. Photophosphorylation. Right. Coming back to here. Where is it? So is it true? So that's why there are scientists in this case here, the scientists who propose the theory that chemical equilibrium, chemiosmosis, is the one that drives phosphorylation, is called Peter Mitchell. So that's the guy who proposed the theory. But the guy who conducted the experiment in order to prove the theory is different person, different team. And the name is, I think, Jagendoft, Andre something. Yeah, this Andre Jagendoft. So what did they do? So they have this isolated thylakoid, the chloroplast thylakoid, and then they have it equilibrated in a pH 4 solution. Okay, meaning that the chloroplast thylakoid, in the beginning, the lumen is not pH 4, but since it is equilibrated in this pH 4 solution, eventually all of these chloroplast thylakoid becoming pH 4 as well. Meaning that this is to imitate the busy sunshine day for a thylakoid. Remember, when the electron transport is so active, you'll get this as well. Lots of hydrogen inside the lumen. Yeah. But this is done without the excitation of reaction center. You see, there's no reaction center or anything. It's just an empty thylakoid membrane. Just that. Right. Then this thylakoid, this acidic thylakoid membrane is transferred to a new solution that has the pH of 8. Then they are left in the darkness, no light whatsoever. However, in this solution, there's also already present adenosine diphosphate and also the inorganic phosphate. If it was true, phosphorylation can happen without light, but happen due to the chemiosmosis, they should get ATP, even without light. So that's what they did. So they leave it for overnight, or a few nights maybe. Eventually, the acidic thylakoid, meaning that the lumen that has lots of hydrogen in it, will try to exit the ATP synthase. When this happens, the rotor head of the ATP synthase will start to turn. As usual, then you will get ATP, right? So that's why we know phosphorylation can happen even without light. Okay, but for plant it's special. It's happening because of photophosphorylation. Okay, so that's the thing that is happening in your mitochondria. In your mitochondria, the phosphorylation is called oxidative phosphorylation. Okay, so it's very important to distinguish how something is getting phosphorylated or otherwise. Okay, what's this? Protomotifos, yes. So the energy available for the ATP synthase, this is called the proton motive force. Okay, meaning that the amount of energy present in the one side of the membrane, creating a potential that can actually drive the production of ATP. Okay. Yeah, I don't want to go too much into this. This is something that you can read later. Yeah, pay attention to this. The name got synonym. ATP synthase is also same as ATPase, is also same as CF not CF1. Yeah. If somebody write CF not dash CF1, that's actually ATP synthase. They're just being specific about it. They're talking about the hydrophilic subunit fused to the embedded membrane subunit of the ATP synthase. Right? But it's actually the same thing. Okay. Right. So you can see that. I wonder if there's a story about how many that gets produced. I think it's in this thing. I think we can print later to give to the rest of the class. Remember I told you about the 3 hydrogen that gets attached? Yeah. Because there is 3 C ring here, the subunit here. There's a reason why only 3 hydrogen that gets attached in this bottom of the protein subunit, because of the protein that is present to receive. There's only 3, right? Okay. I know it's written in here somewhere. Oh, 14. The 3 that we see here, just at the bottom. Okay, it's in this this is why I don't do protein engineering. This is why. This paragraph. Chloroplast, Chloroplasts from vascular plants have been found to have 14 subunits per C ring. What is C ring? This thing. This protein that gets embedded in the thylakoid membrane, the C ring here, resulting in a requirement of 14 hydrogen translocation per 3 ATP, meaning that in order to get 1 ATP produced, 4.66 hydrogen need to get through the tunnel of ATP synthase. Then you get 1 ATP molecule. Okay, so if you talk about the Z scheme, by the time the electron arrives at the end in the NADPH, the amount of ATP that gets produced is 2.67. Because remember, as the electron move up and down in the Z scheme, the some amount of hydrogen that will get in. Okay, this hydrogen, once they get in, they need to go out. As they'll go out, this is the amount of ATP that it can produce. Right. However, this ratio is not optimum because for the Kelvin cycle, which is the second part of photosynthesis, you need 3/2 ratio between the ATP and NADPH to run the Kelvin cycle, meaning that you need more ATP than NADPH to produce sugar in the Kelvin cycle. That is the reason the plant have cyclic photophosphorylation, because more ATP is needed to satisfy this 3/2 ratio. Right? Okay. My head kind of tak boleh nak masuk maklumat 14 subunits ni for some reason, because some books says 15, some books say 17. So kepala aku just tak boleh nak settle down. Tapi dia cakap kat sini 14, kau ingat je 14. Right? Okay. Right. That's the story about it. Yeah. Yeah. Okay. So, and this figure here, it shows you, oops, why it's not? It shows you the membrane with ATP synthase in different organisms. So you have in in purple bacteria, you have in chloroplast, you have in mitochondria. Look at the ATP synthase. They all look the same. All the same. But they are in different organisms, showing that we share the same gene to make ATP synthase protein with these other organisms. Okay? But however, whatever happened before ATP synthase, that's up to the species. It can be anything. In the case of purple bacteria, they have the reaction center. Do you still remember what is the reaction center for the purple bacteria? PS2, what is the reaction center? P1. P68, 0. PS1, P700. Bacteria? P870. Hey. Ada dalam tu. In the web topic here. Ni, web topic number 5. Ada benda ni. P870. We can't see that because that is infrared. Right. So for the bacteria, they have this cytochrome B6F complex. They have the PQH. What's PQH2? Plastohydroquinone. Right. So they have this, and then, of course, with the oxidation of the plastohydroquinone, hydrogen is released into the inner side of the membrane. Then hydrogen will go out, you get the ATP. For the chloroplast, this is what we learned for this whole time. So you should be very familiar with this now. It's a bit more complex because it got to photosystem reaction. Right. For mitochondria, for us human, and also for plant, we have this movement of hydrogen by the action of NADH dehydrogenase. We have the cytochrome B6 complex, we have cytochrome oxidase. All of this activity contributing to the increase in inner membrane chemical potential, right? Concentrated with hydrogen, hydrogen need to exit, you get your ATP. This is what we call as oxidative phosphorylation. Photophosphorylation. There is no photo here. got photo here, got photo here. There is no photo here. However, this one got oxidative. This oxidation. See, oxidase. Yeah, we will learn this. Don't worry. We'll learn this. Right. Okay. Yeah, exam. This is for something for you to do. Have you tried to answer? Very easy. Right. Okay. Now comes the repair and regulation. Okay. The thing is, not only sunlight, any kind of light. Remember, it's a form of energy. Hence the word light energy. Energy, when it is coming more than what you can handle, it can cause damage. You look at all the plants there, especially we live in tropical situation now. Do you see any plants can suddenly apply sunscreen or open up the umbrella? They have to withstand the sunlight that coming to them all the time. Tapi kau tak nampak dah pula, tiba-tiba dia sunburn. Pelik tak? Kenapa kau tak sunburn? Yeah. So, the repair mechanism for this is got why need to repair anyway? Because not all of this energy absorbed is translated into photochemistry. That's the problem. Okay, plant is very efficient, but it's not perfect. There is bound to be some excess energy that do not know where to go. They just stay in there, wreaking havoc. Okay, so before this extra energy wreaks havoc, plants must have strategy to deal with it. So what is the strategy here? So this is the overall pathway, how it happens, okay? That's the title of it. The overall picture of regulation photon capture and the protection and repair of photo damage. So this is the strategy. Number one, the plants absorb the photons, thylakoid membrane, absorb the proton, so many antenna complex. Then the electron get ejected from the reaction center. That's good. You got your photosynthesis. What happened if light is too much? For example, in the noontime, that's very scorching hot. Yeah. Do you like to go under the sun at 12? Stay there until 3? You don't. But you leave your plants right in the middle of the sun. Okay. So, how do plants deal with that? That's what you need to understand. So, when that happens, we call it there is an excess photon. So, first line of defense, plants use the strategy of quenching. Quenching I'm not sure what exactly it means in a simple one word. Just to put off fire. Yeah. For example, I like to drink lemonade to quench my thirst. Yeah. You are depriving of something, then you do something to eliminate the feeling of deprivation. That is quenching. To quenching your thirst, to quenching your loneliness. I don't know. Anything you feel you are in need for. Right. So for this excess proton, plants can choose to do the quenching first. Right. If the quenching takes place, the excess photons will be released back in the form of heat. They just absorb. They use some, but there is a balance. This balance will be quickly released in the form of heat. Actually, that heat is the infrared. You know, infrared, people always associate with heat. Yes, that. So if that happens, no damage is done to the plant. But what if the heat is not sufficient to deal with the excess energy? Yeah, then this thing will happen. The formation of toxic photoproducts. Okay, this is what the plants want to prevent from happening. If they do not know the right way to deal with excess sunlight, this thing will happen within the chloroplast. What are they? 5 things. Triple state of chlorophyll, you have seen that in the second lecture. Superoxide, singlet oxygen, hydrogen peroxide, and hydroxyl radical. I do expect you to know all of these 5 when I ask again later. Okay, and how to properly write this symbol of it. Okay, 3 chlorophyll star, O2 that dots and negative superscript. That dots actually to show it has received hydrogen plus. Okay, singlet oxygen, hydrogen peroxide H2O2, and HO superscript dot, which is the hydroxyl radical. So what if these things are produced in the plants, the photoproduct, the toxic photoproduct? So what? You eat super ring all the time, nothing happen? That is known to be toxic. These things, they how do you say? Number one, most of them kind of are hungry for electrons. Very, very hungry for electron, right? When they're hungry for electron, what they do? Stay still? What? They will steal electron, because remember, electrons need to achieve what configuration? I mentioned this. Electrons in the electron valence, which is the outermost orbit, needs to achieve what configuration? There's only 2 configurations in the electron's valence. Ada thing in the note. I wrote it, no, what? Tak ada pada octet or duplet. Duplet 2, octet 8. Didn't you learn this in your chemistry class? Lepas tu saya buat nak exempted. Sebenarnya tak ingat. Okay, so they try to steal and they get it. What happens? The one that they steal the electron from now will become a toxic product. They will become an oxidant. They will start to steal from the neighbor. So it kind of like a cascade reaction, stealing upon stealing upon stealing upon stealing. Guess what? The whole thing will start to collapse. Okay, until a savior is come. Usually this savior is vitamin C or vitamin with or any substance with extra electron to donate. Okay, for example, like NADPH, that H can be donated. If it can donate, it can stabilize the raging molecules like this. Okay, right. So what if these things have been produced? So the plants do not have any more choice. They have to deal with it. So to deal with that, they start to use the antioxidant. Okay, this is called the second line of defense, scavenging system. Scavenging system means like a cleanup. Okay, you clean up the mess. To clean up the mess, you can either calm down these molecules or neutralize them all together. Okay, so plants use what? Plants use pigments like carotenoids. Carotenoids got there are many kind of carotenoids. Okay, carotenoids is actually a family of pigments. It's not only one thing. Beta carotene is one example of carotenoids. Xanthophyll, zeaxanthin, all these pigments, that color that you know, is a member of carotenoid family. Okay, so they got extra electron or extra hydrogen to give up. They give this up without them becoming an oxidant themselves, like NADPH. Right? They can give up without them causing the problem. Okay, so that's fine. Right? Or yeah, including other components as superoxide dismutase and ascorbate peroxidase. Okay, so if you notice this, ascorbate, whose name is that? Vitamin C, right? Because the science name for vitamin C is ascorbic acid. Ascorbate, that's the thing. Okay, right. So after they have cleaned up the system, usually the system has been damaged. Okay, and the damage always happen to the D1 protein, dimer 1 protein. This should be in the figure 915A, I think. So you can see there is a protein associated with you got your main protein, right? The integral protein, and surrounding it you got your the D protein, the dimer protein. So usually these are the one that gets damaged, and to deal with this, the plants will invoke a repair mechanism. We call it de novo synthesis. De novo means from scratch. They just detach or remove the protein, the D1 protein, altogether. This is not repairing, okay? This is like remove, get new one, put it back. Yeah. And this is a form of, or if you want to remember, a form of autophagy, actually, or chlorophagy, because this is in chloroplast. Chlorophagy. Also, that's the D1. If there is nothing, if the repair de novo do not take place, the D1 will get oxidized. There will be severely damaged, and it will lead to photoinhibition. Photoinhibition is something that you see when plants have been for quite some time under highlight. Then it start to look white. Photoinhibition. Or when you measure the plants, it looks green, but the photosynthesis is low or negative. It has become photoinhibited. Okay, so remember there are two lines of defense: quenching or scavenging. Okay, and another thing that can happen is repair, de novo synthesis. Right? Sometimes the plant just do not want to use this, do not want to use this. They just use this instead. So this is also a line of defense actually. De novo synthesis repair. Okay, it depends on the species. Right. Oh, what's that? Okay, this is actually pretty much what I just said just now. Right. So carotenoids is a photoprotective agent. So that's the word that you need to understand. Photoprotection, kind of like your sunscreen. You know that sometimes sunscreen, if you don't like it this day, you can actually take your sunscreen in pills. Right? Contohnya in a shine. The vitamins that you eat, especially the one that is high in vitamin C, is ACE is plus 2 minerals, zinc and selenium. These can act as an internal sunscreen. Yeah, meaning that if you keep on eating this thing on regular basis, if you go to sunbathing or something, your friends got severe sunburns, you don't get it. Or if you get it, you can heal in about 2 days time. Yeah. So that means you have like an internal shade factor inside of you, right? Not to say that you go to sunbathing every day. Don't do that. You don't have chlorophyll to deal. Your melanin is not meant to deal with that amount of high sunshine. Okay, so when any of this method is used, then the excited state of the chlorophyll is said to be quenched, meaning that the fire has been put off. You've got the fire, and the fire has been put off. But being put off by what? Anything, any defense that you choose, is said to be quenched. Is it damaged? Not yet. Not yet. Okay. It was going to get damaged, but the fire has been put off. Maybe it got some scar to it, but it still can function. Right. Like you got dumped by boyfriend or anything, heartbroken. So you have to come to class and take my exam. Right. Yeah. So you are in the state of quenched. Yeah. Got scar? Scar. Nobody cares, right? Life continues. And some other pigments like xanthophylls also participate in energy dissipation. Right. Okay. Remember the other line of defense that release in the form of heat, this thing? So that is got another name. We call it non-photochemical quenching. Why? If photochemical, that means the electron proceed with electron transfer. That is photochemical quenching. Right. If it's released as heat, we call it non-photochemical quenching. NPQ. Right? Yeah. Something about xanthophylls, actually. I don't know whether it's got in here. Oh, good. It's got in here. Yeah. Something about xanthophylls. Depending, remember I said earlier, the family of carotenoids. They are all carotenoids, but scientists give them different names because they can interchangeable their structure depending on the lighting situation. Here's what it means. Look at here. We take in the case of zeaxanthin. Zeaxanthin is the pigments that usually found in, well, you know any plant with the name zea? Yeah. What color is that? Orange. Yellow lah, zeaxanthin. So zeaxanthin, yeah. That's the very pigment that give color to the corn kernel. Okay, so zeaxanthin actually, so these are all on the surface of the thylakoid membrane. Okay, some part of it got the tail embedded in the membrane itself. Zeaxanthin is present when there's a high degree of non-photochemical quenching. You have a highlight. Imagine the noon time. Yeah, but the plants needs to do a lot of excess heat release. High degree of NPQ. So when this happens, you got your zeaxanthin. So this, you can see here, then it can change to anthraxanthin, it can change to violaxanthin as the light change in color. Why? Because xanthophyll change form due to the pH level. The more as it gets more acidic, zeaxanthin change to violaxanthin. Okay, so by the time it's got violaxanthin, it's actually on the luminal side of the thylakoid already, right? Okay, so you got your zeaxanthin here with the presence of NADPH, oxygen. It will change to an intermediate xanthophyll, which is anthraxanthin, and anthraxanthin will come to eventual violaxanthin. Okay, and during this state, it is low light, meaning that low degree of non-photochemical quenching, meaning that this state, the plant is relaxed. Okay, low light, I don't know, maybe in the sunset, right? Very calm, very calm. When it's very calm, you do the analysis of pigments, you're going to see lots of phyraxanthine. If in the noon you do the pigments analysis, you're going to get lots of zeaxanthin. Okay, but phyraxanthine in the sunset, then the plant undergo overnight. There comes again the sunrise. Then, poof, come again the noon. Can phyraxanthine change back to zeaxanthin? Yes. The plants do not need to make new pigments. They just need to change the pigments into the highlight type. By using what? Ascorbate, vitamin C. Yeah, vitamin C, and then plus with hydrogen. Hydrogen where? Hydrogen that is the high hydrogen that is already present in the pool, because this is highlight. Because it is more light now. And DHA. DHA is dehydro. This is ascorbate actually, but ascorbate without water. I think dehydro something this thing. This is not DHA that is used in your brain. Does it say in here? Dehydro ascorbate, is it? Abdullah? At least I'm not scamming you. Yeah. It becomes anthraxanthin, then use the ascorbate again. You got finally zeaxanthin. When this is present, zeaxanthin is present, it can deal with non-photochemical quenching. So the plants can quench the damaging highlight in the form of heat release. Okay, then comes again the sunset. Get again phyraxanthine over and over. So all of these, we're not going to learn it, but I just want to say this to you. We call it as Xanthophyll Cycle. They are all Xanthophyll Cycle. I don't know. Maybe the biochem students, they learn about this. Right. Right. Okay. The thing that you need to learn here is about the photo inhibition, which I mentioned earlier. Right. So, what needs to be? So whatever story that I said just now is in here. Yeah. Another way the plants deal, well, not really deal, as a strategy to deal with the highlight is the stacking of the thylakoid. You see, when the thylakoid is stacked, what do you call it? The stacking part of the thylakoid. Grana what? Grana lamellae. Grana lamellae. This will reduce the amount of light that gets in the middle. Okay, because the outermost is the one that gets damaged. It's okay. We are in the middle, safe, and can do our job properly, right? So that's why they get the the stacking done in here. Let me see. What's the? Oh yeah, this thing. Let's read together this thing. No single arrangement of pigments, meaning that the antenna complex, would satisfy the requirement to capture the light efficiently, meaning that the amount of energy received by photosystem 2 and photosystem 1 is the same. So that is not happening actually. The plant cannot decide what's coming to it, right? But what they can do is, if the energy is needed in the lesser amount for photosystem 1, they can somewhat rearrange the pigments. So that maybe the journey takes longer for the light to jump, the energy to jump before it reach the photosystem. So let's see how the plants do this. So in order to achieve this efficient light energy distribution, throughout the thylakoid. Remember, some thylakoid are exposed directly to the sun, some are inside, some are actually all the way under, right? So the plants need to decide somehow how to deal with this. This problem can be solved by a mechanism that shifts energy from one photosystem to other in response to different conditions. Okay, we're talking about photosystem now, two photosystems now. Why this can be achieved? Because thylakoid membrane has a protein kinase. Protein kinase is a special protein that facilitates in the process of phosphorylation, meaning that when this protein is around, it can take phosphate from ATP. It gets close to ATP, then it chop off the phosphate from ATP, then it gives this P to the molecule. This is the job of protein kinase that can phosphorylate a specific threonine residue on the surface of LHCII. So in figure 5.15a, you will see that light harvesting complex 2, even though it's kind of in the, I think in the your figure should look cyan, the color, even though it looks like a blob of something, on the surface of it, there is a residue of amino acid. Threonine is an amino acid. Okay. This membrane-bound internal pigments protein, the moment it gets activated, let me see, how can I put it in the correct word? Protein kinase. Protein kinase is actually it's a bit complex. That's why I did teach you, right, 2 years ago. Lepas tu semua tak boleh jawab soalan tu. Who come first? Wait. Protein kinase, go here. It gets phosphorylated. 1 second. Okay. So the kinase, in order for the protein kinase to get activated, plastoquinone, which is the protein in between photosystem 1 and photosystem 2, they need to be accumulated in the reduced state, meaning that for the plastoquinone has get hydrogen attached to its body. This plastoquinone that has received this hydrogen, remember plastoquinone got 3 steps. Plastoquinone, what else? What's the next step? Plastosemiquinone, then plastohydroquinone. Yeah. So when there is a lot of plastosemiquinone and plastohydroquinone accumulated in the thylakoid membrane, one thing will happen. The thing is, it will cause the light harvesting complex to be phosphorylated. In order to make light harvesting complex to be phosphorylated, protein kinase need to be activated. In order for the protein kinase need to be activated, you need to achieve accumulated amount of plastoquinone. See, complicated that thing. It's, I don't want to see, it's a loop. It's like in order for one thing to happen, this thing needs to happen. In order for this to happen, a separate thing needs to happen. But if this thing not happen, this thing not happening. How can I simplify this? Light harvesting complex, it needs to be phosphorylated by a protein kinase. Okay. However, protein kinase itself needs to be activated. For protein kinase to be activated, plastoquinones need to be reduced first and accumulate. So when plastoquinone has been reduced and accumulate, lots of them, it will start to activate protein kinase. And protein kinase, when they have been activated, they will start to phosphorylate light harvesting complex 2, meaning that they keep adding phosphate to it. When this happens, light harvesting complex can start to migrate. It can move. It can move. It can move. It moves where? It moves out of the stack region, meaning that in the crowded region, as you can see in the picture 915, some of them in the crowded region, they go to a more peaceful, not so crowded region, just empty thylakoid space there, into the unstacked region, right? So because of this, they will actually approaching the stroma lamellae. Guess who are in the stroma lamellae? Photosystem 1. So all those extra energy now, because the photosystem 2 cannot handle, they are now channel some of it by the movement of LHCII into photosystem 1. Yeah. So by having this way, surely more and more product can be obtained without having to lose the light energy captured, right? Okay, well, that's the essence of it. Did you get it? Did you get it? Did you get it? Did you get it? I'll just open this thing. 9:15, right? Oh yeah, this thing. Okay. Oops. Okay. So the story is, plants want to prevent photoinhibition from happening. Photoinhibition is a situation when the plants cannot do proper photochemistry due to the excess light present, meaning that the biochemistry of the chloroplast is inhibited with the excess of photons, hence photoinhibition, right? So we know that the plants have employed strategy to deal with this. It employs the strategy of quenching, it employs the strategy of scavenging, or it just let it go and then redo the D1 protein, the dimer 1 protein. Okay. However, the danger is if the plants do not deal with this in the correct way, or maybe all of the strategy kind of occupied, they don't want it to go all the way to the photoinhibition. Okay, because if the D1 protein de novo repair synthesis still cannot be enough to deal with excess energy, photoinhibition will happen. Okay, so plants try really hard not to have this photoinhibition. Okay, so if strategy 1, quenching is not working, scavenging not working, de novo synthesis not working, there's another way to do this. But this way, it involves the detachment of some protein to rechannel the light energy. Okay, meaning that the energy that the photosystems receive, they are trying to balance that, right? If you look at this, image here, most of this configuration, light harvesting complex 2 and also photosystem 2, they are in the middle region here, correct? And actually there are some excess of it, and some of it actually, share the light harvesting complex with another photosystem 2. So very concentrated. So if you look at this, there is no immediate light harvesting complex 1 to it, is that? This guy here, this guy is photosystem 2. This guy is photosystem 1. But there is no photosystem 1 in here. So you're not going to get NADPH from here. Okay, because there is no ferredoxin to receive at the end. So what happens is, plants have this mechanism to move this guy here. This, what color is this? Light purple. LHC2. Move from stack region, this region, into this region, unstacked region. Into this region, unstacked region. Remember, okay, this is very fluid, okay? You don't think that from here can jump into here, but this is actually 2D. The actual thing is 3D. They can actually move. They can move. In order for the light purple LHC2 to move from here to the more uncrowded area, something needs to happen first. What is that something? For the phosphorylation of LHC2, meaning that this LHC2 need to receive energy because it's about to make a hijrah now. Hijrah from unstacking region to unstacking region in its energy. In order to do the migration hijrah, it needs energy. What energy is that? ATP. It needs ATP. Who gives it ATP? Who gives ATP? Actually, it needs the P. It needs to get phosphorylated. But when we say that something is phosphorylated, the layman term for us is actually energized. We want the light harvesting complex 2 to get energized so that it can move from the staking region to the unstaking region. Right. So for that to happen, LHC2 needs to be phosphorylated. And this phosphorylation can only happen when a protein kinase remove phosphate from ATP, put it into LHC2. However, the problem is protein kinase needs to be activated first. And it can only get activated when there is an accumulation of high reduced plastoquinone. The plastoquinone needs to be reduced and it needs to get accumulated. When these two happening, this situation somehow activate the protein kinase. When protein kinase is activated, it can do the job. What is the job? Find ATP, chop off the ATP head, get the PI now, put it into LHC1, LHC2. The moment that happens, LHC2 is phosphorylated, or in other words, is energized. Now it can move from staking region to this region, right? So guess what? The energy now can be transferred to photosystem 1. Yeah, so it's not lost, the excess energy now. If you think about it, this is about the energy management. The plant just don't want to, not only don't want to lose the energy, but don't want to wreak havoc in the membrane. Because if this membrane is compromised, the whole thing is going to collapse, which is a very ugly story. Right? Okay, get it? Faham tak? Susah kan? Susah kan? Siapa boleh cerita balik? Siapa boleh cerita balik? But you get the basic idea. The basic idea. Right. So in order to I forgot to bring my notes. So the essence is, in order to get to understand this paragraph, you need to use two figures now. You need to use this figure, and then you need to use the 915 figure, right? That's why you cannot forget whatever you have read earlier. Okay, this thing doesn't go away. It will keep on haunting and haunting and haunting you until you set. Right? Okay? If this doesn't happen, what will happen? Photoinhibition. This is all to prevent photoinhibition from happening. Okay? Yeah. Life is so much easier, right? If the LHC protein, integral protein, cannot move. Too bad, I can move. Which is good for the plant, bad for you. Right. Okay. That's the quiz. Well, you can do that later. Yeah. Finally. Let's finish this in 10 minutes. The genetic of the chloroplast. Okay, you see where you can find DNA. DNA. What's the full name of DNA? Hai, DNA pun tak tahu. Deoxy. Dia siapa? Deoxyribonucleic acid lah. RNA ribonucleic acid. Nucleic acids, you got two. DNA, RNA. DNA, you got in double helix, double strand, and then helix. It's twisted. RNA, single-stranded. Right. You can find DNA actually in plants, especially plants. I'm not talking about human. You can find DNA in 3 places. Okay. All this while you learn DNA only happen in nucleus. No. DNA actually in 3 places. Nucleus, mitochondria, chloroplast. Okay. From each of these, the DNA got a new name. Nuclear DNA, mitochondrial DNA, chloroplastic DNA. And why is that? Because you see, you learn there are many proteins in the chloroplast, right? Proteins that get involved in the light reaction. These proteins, not only light reaction, light independent reaction as well in the Calvin cycle. These proteins actually, most of them, they use the information to make the protein from the chloroplast itself. Meaning that they use some DNA information in order to make this one protein. Because the protein, you can see that it got many subunits. Yeah. For example, we take Rubisco. I know you have not learned about the Calvin cycle. Let's take Rubisco, which is the primary enzyme in Calvin cycle. Rubisco got 2 subunits, the large subunit and the small subunits. The large subunits of Rubisco, in order to make the protein, the recipe for this, the DNA sequence for this, is actually present in the chloroplast. Yeah. However, the small subunits of the Rubisco, the recipe for this, to do this protein, this information is in the nucleus. So the information from the nucleus get transcribed, and then you get your mRNA, and then that gets translated, and then you got your protein. This protein from the cytosol will be brought into the chloroplast, so that the protein that has been produced already in the chloroplast can fuse together to form a functioning Rubisco. You see, it's not easy to make Rubisco, right? Right. So to say that chloroplast has something what we call as non-Mendelian or maternal inheritance, because when the reproduction involves both male and female contribution to it, you are talking about nuclear DNA. Mom, dad, DNA, half N, fused together to make the new you. So that is Mendelian inheritance. However, because chloroplast and mitochondria, they do not have to fuse with anybody, meaning that they only need to replicate itself during the cell division. These two organelles are purely maternally inherited from the mother's side. And that is what is called as non-Mendelian. That does not have any say in this. Okay, so that's why it's good to see. You wonder, right, when you are in your 70s, in your 80s, if you manage to live that long, how will my energy look like? Look at your mom and grandma. Look at your mom and grandma. That's a very good indication how active or how couch potato you're going to get. Because mitochondria and chloroplast maternally inherited. That has nothing to do with this. Okay? They don't do the crossing of the chromosome. No. That do not happen. Right? Okay. So when this happens, it is important to actually in the case of breeding, but I don't think people pay so much attention to this. It is important to select the female with healthy chloroplasts and mitochondria. Yeah. Sometimes when people do breeding, they just care too much about the high yielding and so on, but they don't pay attention to the vigorness of these two organelle. When you have female with unhealthy chloroplast and mitochondria, the baby that you're going to get, not much gonna happen. Not much gonna happen. Yeah, yeah. So what can be done here? The good thing is mitochondria and chloroplast, since they are independent from the nucleus, because they got their own genetic material, they can mutate on their own freely from the rest of the cell or organism. They can do that, actually. So that's why if you look at your grandma, Ama and Akong are 70 now, but it's already under the bed. It's not even bedridden. And you don't want to be that. You don't want to be that. You don't want your children to be that. How can you do something about it? You can do something from now that to make your mitochondria healthy and more robust by maybe diet or maybe some kind of supplementation or some kind of special exercise. Okay, so that when you reach that age, your mitochondria are not degrading like your grandparents. Okay, so when you have kids, your kids, this is from parking kau tak boleh. You actually pass down the mitochondria that you have worked so hard to correct. Understand? You inherited unhealthy mitochondria. But like, oh, that's too bad. That's not happening so much, right? But you do something. For example, maybe you take a lot of PQH2. I told you, right? There's no supplement for that. I can show you now. I show where. If you want to buy. Aku tak suruh kau makan, okay? I just want to show you this. Yep. This thing. PQQ. I want to get this. Mana ni? Pyrroloquinone, quinone, PTQ. So this is actually a compound that can trigger mitochondrial biosynthesis. Okay, so you take this, and eventually your mitochondria become healthier and healthier. Remember, when you are in the state of healthy mitochondria, and then you decide to reproduce, your baby will get that mitochondria. Yeah. Don't reproduce when your mitochondria is equivalent to your grandma. That's not smart. That's not smart. That's not smart. Don't do that. That has no saying in this. So, somewhat, I'm telling this, maybe somewhat, breeding can have a new paradigm now to look at. Make the mitochondria and chloroplast healthy, and then reproduce the plant, so that the baby inherit all of these vigorous chloroplasts and mitochondria. Okay, right. So I told you about the protein imported from the cytoplasm in order to make a complete protein. The protein components can come from both the chloroplast and also from the nucleus. Okay. A small note on that. What is the name of the small factory that make or manufacture protein in the cell? What is it called? Ribosome. Yes. Therefore, ribosome is not only present in the cytoplasm, it's also present in the chloroplast. You got ribosome again. Right? Okay. Yep. Ooh. Yeah. Right. So chloroplast, what's important in that is chlorophyll. Right. So chlorophyll, we're not going to go into detail into here, but it's worth noting that chlorophyll is a molecule which is not a standalone substance. It's always associated to a protein. Why? Because chlorophyll without protein is very unstable, right? Okay, there are many steps and phases involved in the formation of chlorophyll. However, they are all will come together and form this chlorophyte. Chlorophyte is the porphyry ring that you saw here. When chlorophyte is added with a phytotail, which is the hydrocarbon tail, then you get your chlorophyll. Right. So you can see that there are many phases that come together. Phase 1, phase 2, phase 3. And it all starts with a amino acid. Glutamic acid. Yeah. So that's why it's very important for plants to receive a good amount of nitrogen, because along the process it's going to involve the use of nitrogen to make all of this compound. Okay, you see? Nitrogen here, nitrogen here, nitrogen here. That's why nitrogen is called the leaf maker. Right. What happens when your crop is lacking nitrogen? How does it look? Yeah, yellowish. Yeah. The same thing that you're going to see if it lacks magnesium. Yeah. However, magnesium usually the yellowish is not. It's kind of intervenal yellowing between the vein. Okay. Yeah. And finally, where chlorophyll came from? Well, there is the theory is because chloroplast mitochondria have kind of independent genetic set from the nucleus, it is said that they actually came from the different organism from the past. Some distinct organism come together, fuse together, create a new organism. That is called the endosymbiotic theory. Okay, so for the endosymbiotic theory, there is two lines to it. So I'll just start number one. So in the past, there is an organism that is like your cell, that has a nucleus, that has a mitochondria, but without the energy production. It cannot harvest the energy. It still need to capture the energy somehow from the surrounding, right? But there is a cyanobacterium. Cyanobacterium, it has already got the chlorophyll in it. So this cyanobacterium, actually, they get, what's this? This section here. You know the Pac-Man? What do you call? Phagocytosis. Remember? The amoeba? The amoeba kind of eating, engulfing the small microorganism. So that's happening, right? Maybe it tries to eat. When it eats, oh, you are actually useful. Why don't you stay in here? They diffuse together, they become the first photosynthetic eukaryote, which is the algal cell. So that is the first primary endosymbiosis theory. Okay. The feature is that chloroplast for this organism has double membrane. Double membrane from who? From the cyanobacterium in the past. That's why chloroplast got two membrane. That's why. The other way of symbiosis is because some organisms, especially alga, talking about alga now, they have the secondary endosymbiosis. So we have in the past ancestral cell, like your cell, and this cell. This the first primary endosymbiosis. So this bigger and ancestral cell, they decide to eat up this photosynthetic eukaryote, the primary photosynthetic eukaryote. They eat it up. Again, they eat it up. They're like, you are so useful. We shouldn't digest you. Actually, I don't think that they said that they don't want to digest. They can't digest it very well because this is a working thing. You want to mess with this guy? Not happening. So they kind of coincide together, the phagocytosis. However, the mitochondria in this get degraded because the host cell already have a mitochondria. No need to have so much mitochondria. And then you will have your photosynthetic eukaryote again. However, in this case, the chloroplast have four membrane. Yeah. So when you go in the nature, if you study under microscope, you see that, oh, that chloroplast got two membrane. This chloroplast got four membrane. These are the reason why. All we got is theory. Nobody got the time machine to go back and see this thing is actually happening. All right. Okay, and quiz time. Yeah, habis. Okay, that's one. Jom. Jom lah. So, ni. So this would be your, ambil dulu ni. So if you look at this study question for this entire, because we just finished this chapter, okay? This chapter about light reaction. Penat kan? 4-5 minggu nak habis satu chapter. So look at this chapter. Simpan lebih ni untuk ada yang absent hari ni. You can see that the question actually got a hint where you want to find the answer. Do this exercise. I still want to, you still need to show me where is the answer. I do not know. You know, you can open your, have you opened the file, the folder? You open your folder, you can actually go here. reference here. This is the book that has been scanned. You know, that big, heavy, unfitted book in the my bag book. Oh, perlahan. Senang kan soalan ni? So maybe some of this would be on your final ke? Tak tahu. Kan aku dah cakap, kau buat je belajar benda ni elok. Yang tak dapat A, kalau tak dapat A, it's actually your fault. Things have been given. Tak tahu dah nak bagi macam mana. Saya ada hint lagi nak cari kat mana. So for example, look at the question number 2. What is an action spectrum? What is the relationship between the action spectrum for photosynthesis and the absorption spectrum of chlorophyll? Why would this be different? Okay, the way I want you to answer it, you need to answer it in a proper short essay. One paragraph, don't be crazy about it. However, if there is something from the snippet of the book in here, you can open this. You want to take this section to support your answer, do that. Do that. Look, you need to look like you are trying hard. Yes. Yang orang tak suka kan when people look like trying hard kan. Kau suka kutuk-kutuk orang tu kan. No. I do like people are trying hard so that I can judge you. Alright, okay. Very easy. So use whatever resources that have been given to you. Maybe the information is already in the weekly note. You can take that as well. Don't destroy your notes, okay? You can have the section that you want, have it printed or somewhere to answer all this question and also the MCQ question that I've given earlier last week, alright? And this thing is already present in the folder as well. We will print this and give you this later. I suggest you for to read this, instead of sambil makan kau tengok cerita Korea, apa kata sambil makan kau baca benda ni? You know? Mana benda ni? Ni. Tada. Yeah. So this actually to complement, because there are many fundamental story dalam cerita web topics ni. Alright. Remember, I have decided to reduce the chapter. Right. Instead of 5 chapter, maybe we'll just do 2 or 3 chapter the most. Right. But of course lah, I'll go crazy with each chapter. Yeah. Crazy is default. That's my factory setting. What to do? Right. Okay. Alright. So is that okay? Can you do that? Right? Okay. So we'll start the lesson. Kejap, petang ni, what should we do this petang? Dah tinning ke? Do you have class after this? No, if you don't have class, I mean like if you can go tinning after this, petang ni tak datang. Tinning shouldn't be too long, right? I can finish about 20 minutes, I think. Oh, and check water. Need to do that. Okay, I think that's all for now. Please do the revision. When I expect to see all this exercise done? When is your first mid-sem cuti? Is it when? I don't have the calendar. Tak tahu bila. When? 13? Ni? Okay. Alright. Okay. So, so the target is for you to finish by this week. Maybe in the 20th or 23rd. We'll see. Maybe 20th. I found out that you have committed so much sins. You want to repent, and then you come again on the 23rd. You will make sins for sure. Right. Do that, and that can become your first test. Baik tak aku? Tapi kan, given that perangai student, kau tengoklah, ada je lah benda merepek ni buat. Padahal dah terang-terang ada dalam tu. Let's see. Okay, so is that clear? What to do with your MCQ and essay? What you need to do about it? You look like what? Trying hard, yes. No exception, okay? Everybody do it. I need to make sure that, because there is no group work for this. It's all individual. When the time comes, I'll go to you and see what you have done. Okay? Yeah. Then yeah, and then we can do for some one time more for this for your second test, and we're done with it. All right? Okay. Okay. I think that's all. Any question? All good? All good? All good? Any objection? Regret? Which is too late? Okay, so I'll go to the farm. Okay, I'll see you in a bit.